Okay, so in our previous videos, we've talked about bacteria, we've talked about fungi, and those are, uh, you can at least control those currently by applying either a bactericide or a fungicide. The thing about viruses and nematodes, well, at least the viruses. Viruses, once you get the virus, that plant has the virus and it's everywhere. So, um, you need to make sure that we minimize the infection because once we have that, it's very difficult to get rid of it because it actually takes over the DNA and becomes a part of that plant's genetic material. So we've got this obligate parasite, and it cannot multiply unless it invades that host cell. Then, once it invades the host cell, it is going to interact with that DNA and it will basically like hack the DNA to make more of itself. And so think about this in terms of a computer. Once your computer gets a virus, what happens? Yeah, it's pretty much done. It's time for you to buy a new one. Um, or if not, it takes like you almost have to basically reprogram your computer because the virus has taken over everything. And so the same thing happens in our plant cells. Okay, so the characteristics, we can have either DNA or RNA protein coat. Maybe it's got spikes so that it can get in, and each different virus has a different way that it attacks those host cells. And if you get more into like virology, you'll know more about that. But just to have an understanding that viruses, once you have them, it's very difficult to get rid of them. So we got over 2,000 of these, and about a quarter of them are going to cause plant disease. Some of the different viruses are, tobacco, tomato, or soybean mosaic virus. I want to make sure that you understand one thing about, especially if you're in greenhouses. It does not matter who is coming in your greenhouse if they have like chewing tobacco in their mouth. It's your responsibility to tell them to take that out and wash their hands before they go into that greenhouse. Do not go into a greenhouse with tobacco in your mouth. I'm not saying you, I'm saying anyone. Do not let anyone in your greenhouse with tobacco because you don't know if they have that tobacco mosaic virus on their hands, in their mouth, in their, when they breathe out, whatever. You just don't want to run that risk because once you get that virus in your greenhouse, you're pretty much done. And so we're using greenhouses to grow tomatoes or to grow different plants because our soil is infected, especially up at Oakley Farm. We're growing those in the greenhouse because we've got an infection in the soil. And so you'll have this sort of mosaic sort of modeling uh, that just looks a little different, and you kind of can't get rid of that. So because they infect that DNA, they're going to spread through the whole plant and cause this systemic infection. And because it's in the DNA, it doesn't matter what you apply to that plant, you're not going to overcome that. So those symptoms are going to be that mosaic chlorotic pattern. You might see some necrotic lesions, vein clearing, banding, leafing, and curling, right? So there's this dark green, light green sort of mix in there. It's not very consistent because this virus is trying to attack the rest of these DNA cells. Some of these might, like the DNA in these cells, some of these might be a little bit stronger, but you can see that it's starting to do its job pretty well. We've got some curling. Notice that these leaves are curled, and this is not producing photosynthesis very well. It's just not producing carbohydrates. Just another look at some of our symptoms. We got this mosaic or leaf curl, and you're going to try and come out of this, and you're going to try to apply more water. It could be that this is just like wilt from high temperatures, and it's just difficult to overcome. The plant can't keep up with the environmental conditions. So you would send this off to the lab, or maybe you see some of these other symptoms on your plant that give you an indication that it is likely a virus and um, not an environmental condition. Now, we do have viricides. There is some possible chemical or biological control, but it's very unlikely. We can use crop like resistant crop varieties in that the DNA, we can, like, if we know that we have this susceptibility to tobacco mosaic virus, and we've looked at the structure, it inserts itself square peg round hole in a certain way, right? Square peg round hole. If we make sure that the round hole doesn't accept that square peg, if the virus can't attach itself to that cell and interact with the DNA, then the virus doesn't, and like, it doesn't attack your plant. Make sure that we have virus-free planting material. It's possible that insects could carry these vectors and like viruses as well. And so if we have a piercing sucking, we've got this insect that injects its mouth part into that plant. It's going to move some out, pull some in, and it can be infected with this virus, and you didn't even do anything about it. Like, it's not like you had this. You just had an insect that happened to be around a plant that had this tobacco mosaic virus, and now it got in and infected your whole greenhouse. Like, it can be just that quick. And so, like I said, we're going to use either natural or engineered resistance in crop improvement, maybe something like CRISPR-Cas9 or, QTL, quantitative trait loci, making sure that when, like, where this virus is expected to infect that DNA, that site, you basically just protected that. All right. Finally, moving into nematodes, a couple of quick more slides about uh, the nematodes, in that these nematodes are microscopic worms that are in the soil and very difficult to completely eradicate. So we've got over 4,100 species. They have this needle-like mouth part called a stylet. This stylet right here is what pierces that cell, and they start to basically suck that nutrient out, kind of like a drill, right? We're going to extract that material out of that cell. And these can be important because they're causing, about $200 billion in loss worldwide. Some of the things you'll see are like stunted growth or leaf yellowing. And so if you have the stunt, and we've seen this in most of our other videos, and that stunted growth and leaf yellowing are going to be common symptoms. The sign is whether or not that's caused by a bacteria, or whether it's caused by a fungi or a virus or a nematode. And so for nematodes, it's mostly going to be looking at the roots. Oh, so here's some of our symptoms. We got this yellowing of the plant, and you're like, why is just this little section gone, but this isn't? But we've got just this whole missing area in this field, and it's possible that, this is just caused by an infestation of nematodes right there. But eventually, this section and this section will take out that section. We've got stunting of this growth here. So the plant is still trying to do its thing. It's just not strong enough because we maybe have a soybean cyst nematode that is infecting the plant roots. And if the roots aren't good, then the shoot isn't good. So once you have these nematodes in your field, it's very difficult to eliminate them, in that, they can be moved with water, and especially in soy, like in sandy fields, you have to think about like this microscopic material trying to travel pretty much in the same route that water does, in that you would expect water to move faster in a sandy field than you would in a clay field. So understanding a little bit about your soil environment is also going to help you um, uh, determine on whether or not you likely have the potential for an infestation of nematodes. In between fields, it's possible that you pick up some soil from field A that is infested with nematodes, and then you carry that to field B, and you've now infected both field A and field B. So making sure that your equipment is clean or that you don't have an infection or you don't have these populations is going to be important. We have four types of nematodes. We have root knot, cyst, lesion, and reniform. So as we move into these next couple of slides, just to make you aware of the different types, what do you think we're going to have in our root knot? Probably a knot on the root, like a gall. For our cyst, what do you think we're going to have on the roots? For our lesion, and then for our reniform. All right. And so, not that I care that you know too much about these, because if you get into this field, the more that you interact with this, the better off. Like the more experience that you get with these different types will also help you combat them. And I think what I'm going to do in module 3 when we go over these, because we'll be using like cdms.net. And once you know what type of nematode that you have, you can then choose what type of nematicide that you want to use. All right, so our root knot is going to be the most important one. Again, we've got 100 species worldwide. They're going to attack a large variety of hosts. Pretty much if you're growing a plant in a field that has nematodes, that's an opportunity for that nematode to destroy that. So soybean, cotton, and corn, our vegetables and our fruits, we're going to have roots growing in the ground that are close to uh, the surface. And once it attacks those roots, then our shoots or our fruit plants above ground are not going to perform very well. And this is pretty much for all of our nematodes, right? Like you'll see in the next couple slides, the soybean, cotton, and corn are definitely susceptible to nematodes. So, these juveniles are going to attack the root tips because that's the most uh, that's kind of the newest material that hasn't really hardened off yet. It's very soft, easy to penetrate, and it's also it doesn't really have like a big armor. And when I say armor, I want you to think about this in terms of like how many layers of cells before you actually get to the xylem. That's what I mean when I say like they're easy to get to or it doesn't have enough armor. Those cells haven't thickened off enough that they can withstand that little small stylet. So here is our gall caused on this tomato root. And again, you got to see this under a microscope. So here is this gall, this sort of infection that is sucking all of those nutrients out. And then we have these galls on okra. So we've got wounds, we've got loss of nutrients, and if we have wounds, then we have more opportunities for those pathogens to get inside of that plant and infect that. So not only is the root knot nematode doing up like killing your plant, it's also opening up the opportunity for other pathogens to make their way in. Next, we have cyst nematodes. And so different from our galls, we're going to have these little knots, like these little cysts that are on the root. Much different from the galls, right? Like this is a large structure, relatively large, considering it's a root, compared to these little cysts, these little sort of pustules that aren't supposed to be there. So we have soybean and potato cysts are going to be an issue. And think about potatoes, like we sort of plant those underneath the ground. And where do you think the nematodes are? Exactly. So, soybean cysts can cause, about $1.5 billion worth of loss. Our potato cyst can, reduce yields 50 to about 80%. And essentially, what this is this cyst nematode is the dead female body with those eggs. And so once those eggs mature, this cyst will open up, it will pop, and it will um, increase the number of nematodes that you have now. So you don't know how many nematode cells are in these little cysts, but it's an opportunity. It's an energy source for feeding those eggs. Next, we have lesion nematodes. They in about 70 species, causing about 85% of crop yield loss. And so you'll have these sort of darkened lesion areas. And if you recall, we can have this could be, mistaken for some bacterial disease. So not only would you send in the plant, but you also might send in a soil sample. And so they would determine if how many nematodes that you have in there that are causing these lesions on this. Like, do you have the sign? Here is the symptom. We've got these lesions. What is the sign? Bacteria or nematode? And then finally, we have reniform. So our reniform nematodes, you're not going to be able to see them. They're not going to cause a gall or a cyst. They're just going to cause an issue, and you won't know that you have that. So these you'll definitely have to send in the plant and the soil sample in order to determine if you like which nematode that you have. Economically important crops, again, soybean and cotton for us, uh, maybe pineapple, sweet potatoes, cassava, etc. Again, if you've got a plant that is going to be like grown in the soil and you have nematodes, you're going to have an issue. All right, so that ends it all for module 2.2. Remember that we went over bacteria, we went over fungi, viruses, and nematodes, in that we have a symptom, something is wrong. We need to determine what is causing that so that we can take the appropriate action. And as we move into module 3, we're going to learn how to, we're going to learn different mechanisms and different methods of control, sort of an IPM thing, and then also look at how you go about finding this. So I don't expect you to know I don't expect you to know that if you have a reniform nematode that you need to use this particular nematicide. I'm going to show you how to go find the nematicide based on what you believe is your issue. All right, that's all I have for this. We'll see y'all in module 3.